All right, last part of chapter 4, looking at DNA replication and the cell cycle. So how do we create more cells for our body? So we'll go through DNA replication, the cell cycle, and mitosis. So in order to have two new cells, you must have two copies of the DNA. So each cell has every single piece of gene coding for every single protein that you might need in the new cell. So in order to make sure that you have the correct copies, you have to be very exact on replicating of your DNA strand. So going back to the law of complementary base pairing that we talked about earlier, where your one strand of DNA is going to determine the pattern of the other strand of DNA. So your nucleotides are always going to pair with each other. Always adenine with thymine, always cytosine with guanine. So we have our big, long, giant strand of DNA. So in order to get to our individual strands, we have to unwind the DNA, and we have an enzyme for that. And that enzyme is DNA helicase. So that enzyme, ACE for enzyme, is going to unwind the DNA double helix. So open it up so that we can get to those single strands, and those single strands are going to determine what the new partners are going to be. So in this area of our DNA opening, it's called the replication point, and that's where we're going to start our copying of our DNA. So in comes a new different enzyme, DNA polymerase. So again, ACE for enzyme, and its job is going to polymerize the nucleotide so we create a new DNA strand. So as our DNA polymerase goes along our original strand, which is the strand you see in blue, it's going to pair the correct nucleotide to the correct partner. So again, thymine is always going to pair with adenine. Guanine is always going to pair with cytosine. So we have two strands of DNA. We have two DNA polymerases working in opposite directions of each other. So one's going to go towards the replication fork, one's going to go in the opposite direction. Because of this going in the opposite direction, again, that's one of those evolution situations, I have no clue why. You'll have one strand where it will just continuously build a new strand, you don't have to worry about it. But the one that is going away from your DNA replication fork only can go so far before it kinks up. So here you have your old blue strand of DNA. It flows in the shape of this DNA polymerase protein. Here you're not flowing correctly. You can work a little short piece, and then if it would have kept going, it would kink up the DNA. So this DNA polymerase going away from our replication fork only does in little short segments, and then you have yet another enzyme that has to come back and fix these little fragments. So they're going to close the gap in between the fragments of DNA that was created by the DNA polymerase, and that enzyme is DNA ligase. So they go through this whole process of building new strands of DNA based on the old strand of DNA, and that is called semi-conservative replication. So each daughter strand, so the new strand of DNA, is based on the old one. So using those law of complementary base pairing, partnering up the same nucleotide with the same partner. So that's just the whole picture of everything. So our one strand being built toward our replication fork, our other strand being built away, and our DNA ligase putting the fragments together. Okay, so now we have our new set of DNA. We have to go through the same process, get it all organized, so basically it doesn't do this inside of the nucleus. So just like your headphones get all tangled up, we don't want our DNA tangled up. So during the process of DNA replication, those histones, you got to make a whole new set of those. So we got a lot of protein production going on. so that we can create those nucleosomes. We wrap part of that DNA around and link them together, and then we fold it back up, and then we fold it even more so into chromatin, which is the last level in a non-dividing cell. But in this section of chapter 4, we are going to divide the cell. So we go into our last final packaging of DNA in a dividing cell, which is our chromatids. So we have our sister chromatids, our paired copy. So again, one pair from your mother and one pair from your father. So our 2 strands, and they're going to pair with each other based on they are the copy of each other. Okay, so we got our new DNA. Sometimes there's mistakes. I mean, hey, it's life. During the process of our cell cycle, getting everything prepped so we can make a new daughter cell, there is a phase where the DNA is checked. So let's go back to our DNA pairing. Maybe. Do I have a piece here? There we go. Okay, let's say hypothetically we have a thymine that was put here where there should have been a cytosine. Well, because that doesn't match up. So remembering our hydrogen bonds between our nucleotides, when that happens, it kinks out of our DNA. So you end up with, instead of a nice, pretty, smooth DNA as it's being checked, there's a little kink out of your DNA strand. The bonds aren't fitting the way they should. And that way you're able to go back and be like, oh, oops, my bad. DNA ligase can come through, stick in the correct nucleotide, and fix any possible caught mistakes. Occasionally, sometimes you keep the mistakes, either just because something wasn't found and you end up with what's called a mutation, or because you had an original nucleotide that wasn't correct, that wasn't fixed, and so when you made your copy, the error repeated. And sometimes that's good. You can actually make, let's say, for example, like a bird. And the new mutation ends up with the bird having a longer beak, which makes it better for it to feed. So some mutations are good. Some can possibly kill the cell off, as in no longer being able to correctly make a protein that is necessary for function. Some mutations can cause the cell to turn into a cancer, so the growth regulation of that cell no longer is functioning correctly, and so you just have this abnormal growth, a bunch of cells they're not working, they're just keep growing and making a bunch of new daughter cells. They're not helping out the tissue, they're just growing, sucking up the body's resources. Or it can cause genetic defects in future generations. So for example, when you're making of your haploid gametes, so your sex cells, your sperm and egg, when that happens, whatever mutation happens occurs in the new generation if the egg and sperm meet in that situation. Okay, so we already talked about how we get new DNA, which is down in this phase. So let's talk about the rest of the phases of the cell cycle. So the cell cycle has this big chunky phase called interphase, which has 3 sections inside of it. So we have our G1, which is our first gap phase, S phase, where we have new DNA, and G2 phase, which is where we get the new organelles. So our new daughter cell as well as the old parent cell both need all the little mini factories that are hanging out in the cell doing their job. Both cells need their own set. So we have to have new organelles, new cytoplasm in order to create the new cell. Then the last phase is the mitotic phase, also called mitosis. It also has little phases inside of it. So we have our prophase, metaphase, anaphase, telophase, and cytokinesis. So this is the phases that you actually can see under a light microscope. So if you want to see that in the lab, just let me know and I'll find the slides. So this is where you actually can see the DNA because it's compacted enough, and then they have specific arrangement as they are organizing everything so that you can split everything and finally get to your last daughter cell. Okay, let's go through interphase. So interphase has these 3 subphases in interphase. So G1 is called the first gap phase. So this is just normal cell growth. We're not preparing to go into creating a new cell. This is just your smaller daughter cell growing and becoming larger, filling in the space, filling in that gap that was open so that you could have a new daughter cell. So that's just your first growth phase. Sometimes your cell will enter what's called G0, where it'll just stop growing because you don't need a new daughter cell. And so instead of shifting to S phase, preparing for a new cell, you just stay in G0 until you actually need to split. Okay, S phase, synthesis phase. So this is where we get our new set of DNA. S phase, new set of DNA. So this is where your DNA is copied using the law of semi-conservative replication, one strand determining the pattern of nucleotides on the other strand. So once you have your new set of DNA, then you move into the last subphase of interphase, which is G2 phase. It's our second gap phase. So another growth phase, we're getting new organelles. If there's anything that happened in S phase where we have an error in our DNA copying, that's when that's going to get fixed. And we're preparing everything ready to go before we enter into M phase, which is also called mitosis or the mitotic phase. So this is where we're actually going to form our new daughter cell. So here at the beginning, we're still one cell, so we're getting ready to eventually split into two. Okay. So during this phase is the only one when your DNA is packed as chromosomes, so that X-shaped looking DNA compaction. And in your M phase, you have, our different phases that we'll talk about later. Oh well. Okay, so purpose of mitosis. It's one of your study guide questions, by the way. So the result is two genetically identical daughter cells. Reason why is for one development. So we go from being a one fertilized egg into trillions of cells. So we need new cell after new cell after new cell to create what eventually becomes an adult, if you want to claim being an adult. Okay. Everything has to grow. We go from a small baby to a larger human. So you need more cells in order to do that. As well as, for example, if you start weightlifting and you're putting that stress against the muscles, you'll end up creating more muscle cells, and so that's also going to be using mitosis. When a cell dies off, so some of our cells are, particularly the ones exposed to an outside environment, they're going to get rubbed off, they're going to die off because they get damaged, so that opens up a space for new daughter cells, so you go through mitosis. And then anything that is damaged. So you have a cut on your hand, you're going to build new cells, at least on the epidermis of your skin, and that's going to use mitosis in order to create those new skin cells. So for mitosis, we have 4 phases: Prophase, Metaphase, Anaphase, and Telophase. Easy way to remember that is PMAT. So prophase, that's the first phase. Everything is condensed in our nucleus. We have our chromatids. So you'll also again hear exchange called the chromosomes. So a little X-shaped DNA, you can see it now under a microscope. Our nucleus starts to dissolve, so that nuclear envelope around it, so that you can get everything out in the open. We have our mitotic spindle starting to form using the microtubules of our cytoskeleton. Oh, go back. So down at the bottom is what it looks like under a microscope. So able to see all that DNA in the middle, little fuzzy centrosome at the bottom where you can start seeing the mitotic spindle starting to form. Okay, next phase, metaphase. Everything's aligned on our central equator. So all of your DNA, you'll see it in the middle. So basically, get off. All of your DNA is just being organized. Let's put it in the correct spot. So before we start dividing, we divide everything down the middle. Everything is organized, so we'll make this a nice clean break. By this time, our mitotic spindle, which is lemon-shaped, so kind of an oblong type of shape, that's completely formed now. And it's time to start splitting stuff to the two sides to create our new daughter cell. So that's both of them together. Okay. So the next phase is anaphase, and this is where stuff is starting being broken apart. It's also the beginning of cytokinesis, where we're also moving the rest of the cytoplasm to your two sides. So here you can see your sister chromatids, which were in that X-shaped, they're being pulled apart. So one side is going to one half is going to one side to a new data cell. The other half is going to the other side. So making sure both of them are going to have complete sets. So remember, by now you have two whole sets of DNA. So we're just splitting everything down the middle. Nice, clean break to get everything that each cell is going to need. So this is where we use the motor proteins on our microtubules to crawl along those microtubules and move everything to the two separate sides. Last phase is telophase. So this is where we have a new nuclear envelope starting to form. We have even further movage of cytokinesis. By now you also see what's called a cleavage furrow. So it looks like cleavage. So you're pinching off of that plasma membrane, starting to actually look like two different cells. Your chromosomes will uncoil to chromatin, so chromatin being the highest level of compaction in a non-dividing cell. Nuclear envelopes forms again. So those two steps all put together just in case you wanted to see them together. Okay, then we're going to go to cytokinesis in a second, but to get these first four, a little memory trick, again going back to our PMAT, because you will identify this for the test. So P, pop open, so we're popping open our nucleus. M phase for our metaphase in the middle, so all of our chromosomes are aligned in the middle. Go away. So we're splitting our chromatids, so our sister chromatids going to our two different sides. And the last one, telophase, back together. So putting everything back together, put our nucleus back together, splitting everything, getting everything organized to our final phase where we clip off, and that is cytokinesis. So our last final division, cleaving off of the two cells, so they're now two separate ones. So in the identification on the exam, I'm just going to do a close-up of where we have this cleavage furrow showing the cutting off of the two cells. So everything is finally separated, and we're just cleaning off, making a smooth break. Okay, so why do we have cells? So of course this is regulated unless you have a tumor. So cells only divide if there's enough cytoplasm. So you have to have enough stuff for both cells to form. Now there is a slight difference to this. We'll talk about this in the female reproductive chapter, which is chapter 28. So basically AP2 material. But there is some differences there where actually more of your cytoplasm is reserved for one cell, and then the other cell that doesn't get the cytoplasm is actually thrown away, but that's okay. Normal cell division, it's having enough cytoplasm. Another one is you have to have two sets of DNA, so you got to get through that S phase. Making sure there's enough nutrients. So if there's not enough nutrients, that new cell is going to die off, so there's no point in making a new one. If there's something with a growth factor, so kids, they have those growth spurts, it's because they're given the signal to grow. So that post-kindergarten growth spurt, that puberty growth spurt, and then some, particularly males, because they tend to grow for longer, like just continues until almost adulthood pretty much. You have cell division when there's open up space. So you have a damaged cell that dies off, or you have a cut. So now there's more space. And so the cells divide to fill that gap in. So cells stop dividing when they make contact with neighboring cells. That means there's not enough space to keep pushing stuff out the way. So you stop making new cells. If there's no growth factor, so that kindergarten growth spurt, they grow a little bit tall and then they stop. So when those growth factors are not there anymore, they're not given that signal to go through mitosis. Okay, so this is the last slide for chapter 4. Again, on your exam, you will identify the different phases of mitosis, including cytokinesis. So we have our prophase, metaphase, anaphase, telophase, and then finally cytokinesis. All right, if you have questions, let me know.